Okay, thank you. Thanks for the invitation. Also to the organizers who know better than anybody else in the room, thanks for putting up with all the hassles I've created in the process of showing up. It's my pleasure to talk to you about some of the work that we've been doing in my lab at the NICHD. The lab from its inception has had a fundamental focus on the study of individual differences in personality or temperament in the Rhesus monkeys that were able to maintain in a fairly large colony out in rural Maryland. We're interested in these individual differences in the biological substrates that underlie these individual differences and what the long-term consequences are with respect to health and well-being. For example, we've found over the years that about 20% of the monkeys in our colony, which is near a little town called Poolsville, so I'll refer to it as the Poolsville colony from now on. About 20% of the monkeys in the Poolsville colony, as well as about 20% of the monkeys at 2 field sites we've had access to, seem to be unusually nervous and anxious in the face of novel or mildly challenging circumstances. They also show a number of biological features that differentiate them from others in their group. The feature we spent the most time focusing on is the HPA axis, and these monkeys, at least early in life, under comparable circumstances, have higher and more prolonged elevations of cortisol, whether we're measuring it in blood or saliva, or more recently in hair, than those of similar age and background. These monkeys are at risk for developing anxiety and depressive-like disorders when studied in captive circumstances. It's another 5 to 10% of the population who seem to be unusually impulsive, and this impulsivity gets them into trouble. They do stupid things that other monkeys know better than to try, like get between a high-ranking female and her offspring, or they may repeatedly confront a dominant adult male, and when that male beats them back, they're right back in that male's face, and when that male beats them back again, they're right back in that male's face. So not surprisingly, they tend to elicit a lot of aggression from others in their group, and they themselves are excessively and often inappropriately aggressive. And more than that, once they get into an aggressive encounter, whereas most monkeys know how to back off and calm things down, when these monkeys are involved in an aggressive encounter, often the intensity of the aggression escalates to the point where somebody can actually get hurt. And these monkeys also have biological features that differentiate them from others in the group. The feature we paid the most attention to is an apparent deficit in serotonin metabolism that we've been monitoring primarily by looking at levels of the primary central serotonin metabolite, 5-hydroxyindolisidic acid in the CSF, and these monkeys have chronically low levels of CSF5-HIAA. As I mentioned, my lab is not on the main Bethesda campus. It's located in rural Maryland near the Potomac River, about 40 miles up the Potomac from downtown DC, 28.6 miles from the main NIH campus. Here's an aerial view. Here's the Potomac River. This is Virginia. Look at all the houses and parking lots and stuff. This is Maryland. Look at all the green. If you ever believed in the importance of zoning laws, there you go. We could never have this lab over there. Here's the outline is the boundary of the NIH Animal Center. It's about a mile on each side. And in red is my lab. We're fortunate to be able to measure lab space in terms of acres and not square feet. Within the lab, within the area, is a field station of 5 acres with a sheltered area and a pond. Here we can have monkeys living in free-ranging groups that mimic what you see in their natural habitat. And in their natural habitat, these monkeys typically live in large social groups ranging in size from 30 or 40 individuals up to several hundred. But whether the groups are large or small, they all have the same basic social structure. Every group is organized around several female-headed families or matrilines, plus males who come in from the outside. And this arrangement derives from the fact that females stay in the group in which they're born for their entire life, whereas males stay only until puberty. They leave home around puberty. They typically join all male gangs, and then they, after about a year or so, they try to work their way into a new troop. So every troop has plenty of adult males and adult females in it, but the females have all been there since birth, and they're all related to one another, whereas the males have been only since puberty. And initially they're related to no one until they start having offspring of their own. This species is not our closest biological relatives. I don't have to tell you people this. They only share about 95% of the genes that we have, as opposed to 98 or 99% of that chimpanzees and bonobos share with us. But I can argue that I believe rhesus monkeys are more like humans than our chimpanzees or bonobos or any of the great apes in one fundamental respect, and that is they are a success story. They are the world's second most successful species of primates. Next to us, there are more rhesus monkeys living in the wild over a larger geographic area, experiencing a wider range of climates than any other species of non-human primates. And in the Indian subcontinent, where they come from, you can find them everywhere from rainforests to savanna areas to the edges of the desert, and even into the Himalayas, up to 13 or 14,000 feet, where the winters are a lot more severe than anything around here, which is why even in the worst winter in Washington history, our monkeys did just fine in the field station. Here's that pond you saw before. It's now frozen. But there are monkeys out in the sun getting their rays. When they get a little cold, they run back into their shelter and then go back out into the sun. They are extraordinarily adaptive. And also, one last thing about rhesus monkeys as a species, they are a weed species. They're one of the very few weed species that you see in primates. That is, if a weed species is, if you take that a group out of their natural habitat and put them someplace else, weed species are likely to thrive and expand their population, whereas most others and most other primates wouldn't last very long. And as one example, this is from a newspaper about a month ago in Gainesville, Florida, close to a place called Silver Springs, where 80 years ago they filmed the first Tarzan movie, and in that particular habitat. And they brought in some rhesus monkeys for background to make it look more like a jungle. And when the filming was done and the actors left and the producers and the directors and the camera people left, the rhesus monkeys stayed around, and they're still there. At least their descendants are still there 80, 85 years after that point, despite major efforts by the farmers to get rid of them. They consider them agricultural pests. The rhesus monkeys are too smart and adaptive, and I think they're going to be there as long as the people are there as well. So this is a highly adaptive species, and I'll get back to that point at the end if time permits. Now, as I said, we know a lot about these monkeys as they grow up. More than 50 years of data, including Jeannie Altman's husband, who did some of the original work, Caio Santiago, many, many years ago. As I said, these monkeys live in large groups, social groups that are organized around multi-generational families. Here's one of them. Here's the mother, newborn infant, grandmother's back there. This is the older sister of the infant. This is the younger sister of the mother. And the second thing you should know about rhesus monkey social organization is that they have multiple dominance hierarchies. And these hierarchies are, relative to other macaques and most other primates, exceedingly strict and unyielding. There's a hierarchy between families, such that every member of the highest ranking family, including infants, outrank everybody in the next highest ranking family, including adults. So you have situations in the wild where infants from low-ranking families may be stumbling along a trail, and adults from high-ranking families will be falling all over themselves to get out of the way of these little kids, because they know if they cause these kids any grief, the rest of that high-ranking family will be on them in a moment. There's a hierarchy within each family. Younger daughters outrank older daughters. There's a hierarchy among the males that come into a group, roughly related to tenure or how long they've been there, but of course it's not length of time that's important. It's how good those males are at making friends and sustaining alliances, not only with other males, but especially with high-ranking females who carry the real clout and the truth. And males that are good at that stick around and move up their hierarchy, and males that are not so good give up, leave, and seek their fortune elsewhere. And finally, there's a hierarchy among the infants that are born into a group every year. These are cesto breeders, so all infants are born at roughly the same time each year. And the position in the hierarchy of these youngsters reflects the position of their mother. So it's not that some infants are bigger or stronger or smarter or quicker than other infants. It's that other monkeys associated with a particular mother and afforded the equivalent social status. So life in a rhesus monkey troop is a pretty complicated business for individuals to survive, let alone thrive. Not only must they develop a complicated behavioral repertoire, but they also have to learn a great deal about others in the group. who's related to whom, where does each individual fit in these multiple dominance hierarchies, as well as recent social history. Who's been getting into fights recently? Who can you count on to back you up if you get into a fight yourself? And monkeys that are able to assimilate this information, follow the rules inherent in these very strict dominance hierarchies, do very well. And those who are either unable or unwilling to follow these rules don't survive very long, at least in the wild. A few words about basic development, because that's what a major focus will be on. Rhesus monkey infants, who by the way grow up 4 times faster than we do, which is an enormous advantage for those of us who are interested in long-term effects of early experience or lifespan development or a transmission of characteristics from one generation to the next. With reference to that early human paper that we heard earlier, we can see a generation in 4 or 5 years instead of having to wait 15 or 20. So when I give you an age in rhesus monkey months or years, just multiply by 4 and you'll get a rough human equivalent. At any rate, these infants spend virtually all of their first month of life in intimate social contact with their mother, and during this time, a strong and enduring attachment bond is established between mother and infant. It's a bond that's functionally equivalent to what human infants routinely form with their caregivers in virtually every culture in the world. And once this bond has been formed, infants begin to leave their mothers for short periods of time to explore their environment. They use their mother as a secure base, will wander away from their mother for a short period of time, then go running back to their mother for a period of contact, explore a little bit more, back to their mother for a period of contact. It's almost as if the infant were attached to the mother by a long rubber band. And as the infant gets older, the time spent with the mother becomes less and less, and time spent away at greater distances and for longer periods of time increases to the point where by 6 months of age, these youngsters are spending only about 20% of their waking hours in actual physical contact with their mother, as opposed to the 100% that they were spending in that first month of life. Yet the mother's presence in the immediate environment is absolutely crucial to sustain this early exploration, because as Bowlby taught us, if you lose access to your secure base, any motivation to explore will disappear quickly, and you'll become emotionally upset. So even though these youngsters are not spending that much time with their mother at this point, their mothers still remain an integral part of their social world. Who do they spend their time with? Well, increasingly they spend their time with peers. As I mentioned, these are seasonal breeders, so all the monkeys born each year are born at roughly the same time, and that means that monkeys born during that period will have plenty of others, monkeys who are the same age, that have about the same physical and cognitive and social capabilities as themselves, and that's who they choose to hang out with. So throughout the childhood years, the rest of the first year of life, the second year of life, and the third year of life, these monkeys will spend several hours every day in active social play with their peers. And it's in the context of this play that virtually every behavior pattern that's going to be crucial for normal adult functioning can be developed and practiced and perfected long before it actually has to become functional. And this is especially true for the socialization of aggression, which comes into their repertoire between 4 and 6 months of age. Now, not all monkeys out at Poolsville grow up this way. Some of them are cross-fostered to unrelated females at birth, so we can separate genetic and environmental factors. And others are reared according to a procedure developed by my mentor, Harry Harlow, more than 50 years ago, without mothers, called together or peer-only rearing. Here monkeys are separated from their mothers at birth before any kind of attachment relationship can be developed. They are hand-reared in a neonatal nursery for the initial weeks of life, and then at about a month of age, they're put into small groups of 3 or 4 or 5 individuals who were similarly separated from their mothers at birth, and then they live together 24 hours a day, 7 days a week, until they're about 6 or 7 months of age. At 6 or 7 months of age, they are moved into larger social groups that contain their mother-reared counterparts, who at that point are weaned from their biological mother, just at the point where monkey mothers in the wild actively wean their own offspring to get ready for next year's infant. So from 7 months on, mother-reared and peer-reared monkeys are living in exactly the same physical and social environment. And so any differences that we see in their subsequent behavior or biology, we can attribute to that first 6 or 7 months of life. And when you rear monkeys this way, they become hyper-attached to one another. Very strong attachments, but these attachments, while very strong, are actually dysfunctional because these monkeys spend excessive amounts of time clinging to one another instead of exploring, beginning to explore their larger environment. And when they get to the age when they should be starting to play, they do develop patterns of play, but the play never reaches the level of sophistication or intensity or frequency that you see in their mother-reared counterparts. So even though these monkeys are with potential playmates 24 hours a day, they're actually play deprived as they're growing up. And we now know that this has consequences. For one thing, several decades of work has shown that monkeys so reared quickly become excessively fearful or anxious in novel situations. In that respect, they look exactly like that 20% of monkeys in the wild that I described earlier, and like that 20%, they show excessive HPA activity. They also, as they get older, start developing these explosive patterns of aggression that you see in that 5 to 10% of the population in the wild. And like that 5 to 10% of that population in the wild, they show problems with deficits in serotonin metabolism. Extensive laboratory research during their juvenile years has shown that they also are very poor at delay of gratification tasks, and they have other cognitive problems. They have higher levels of C-reactive protein. They have more immune function difficulties. When my colleagues in the Alcohol Institute put them in a happy hour situation, they consume more alcohol than their mother-reared counterparts. And more recently, we've been able to do some neuroimaging studies of these monkeys, and their brains are different. These are PET scan data. I'll just walk you through the left side, looking at serotonin binding potential. This is a composite, front view of a composite mother-reared versus peer-reared, mother-reared versus peer-reared, mother-reared versus peer-reared, at about 2 years of age. Again, living together for 18 months after that initial early experience, and this simple inspection suggests that the pure-reared monkeys' brains are lighting up more than their mother-reared counterparts. And you look at the actual data, and indeed that is the case throughout the brain, whether you're looking at the raphe, the thalamus, the striatum, the temporal or the frontal regions. Pure-reared monkeys have consistently lower serotonin binding potential than their mother-reared counterparts. We recently published a paper looking at another PET scan study looking at serotonin 1A receptors. We get the same kind of rearing condition effect. We have recently published a paper looking at structural MRIs. Their brains differ in several fundamental areas as a result of this early rearing. So, and most recently, we started to look at genome-wide gene expression and methylation patterns. I'll spend most of my minutes talking about gene expression studies. These are data collected, are analyzed by Steve Cole at UCLA in a collaboration that Dr. Sussman arranged some years ago with Jim Heckman at Chicago, myself, and Steve Cole. And, oops, sorry. The other thing I was going to mention is we've been looking at gene by environment interactions, looking at candidate genes as they might interact. I'll just briefly talk about the serotonin transporter gene, for which rhesus monkeys and humans have the same polymorphism, that due to variation in the number of base pairs in the promoter regions, such as the long and the short version of the gene, and in both rhesus monkeys and humans, it's a functional difference. The short allele is associated with less efficient RNA transcription. And when we look at whether it makes a difference, if these monkeys have the long or short allele, it does, depending on how they are reared. If they're pure-reared and they have the short allele, they have a deficit in serotonin metabolism. But if they were mother-reared, it doesn't make a damn bit of difference which allele they have. They have perfectly normal serotonin metabolism. This is a gene by environment interaction. When you look at aggression, you get exactly the same picture. Short allele, pure-rearing, high levels of aggression. Same short allele with a good mother, as lower or as low levels as mother-reared or peer-reared with the same allele. And I could give you 8 or 9 other sets of dependent behavioral and biological functions that show exactly the same pattern. We see it, well, here's alcohol. Here you get a reversal. Peer-rearing with short allele, monkeys drink to excess, but same short allele. Mother-reared, they actually drink less than normal, so what may be a genetic risk factor for individuals with poor early experience may actually be a genetic protective factor for individuals with good early experience. And we see the same pattern in the 8 or 9 other candidate genes we've looked at. There's MAOA, where Rhesus monkeys and humans have structurally different but functionally equivalent polymorphisms. There's a relationship with aggression that mimics the pattern that Caspi and Moffat found in Dunedin. With individuals with a less efficient version showing higher levels of aggression, but only if they also had a history of child maltreatment. And here's a SNP and a dopamine, a SNP and a CRH receptor looking at alcohol for Stephen Barr's data. And again, the same pattern. Pure rearing with the less efficient gene is associated with high levels of alcohol consumption, but the same gene is same allele, but with good mother rearing, lower than normal levels. So the pattern, as I said, adverse rearing condition paired with less efficient allele has negative outcomes, but that same less efficient allele in the presence of good mothering yields normal or better than normal outcomes. I started to talk about gene expression. We're going to have to go pretty quickly. So here are Steve Cole's data. This is a heat plot. Every vertical line is a gene. This is genome-wide, looking at leukocytes. Each horizontal line is a subject. Mother-rid, 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 pure-rid, pure-rid, pure-rid. If you see red, the gene is overexpressed. If you see green, the gene is underexpressed. And what you see is virtually no overlap as a function of rearing. If it's red with mother-reared, it's green with pure-reared. If it's green with mother-reared, it's red with pure-reared. And you might say, that's nice, but what does it mean? Well, Steve Cole knows what it means, and he says that the genes that are underexpressed in pure-reared monkeys, immunoglobulin production, interferon, antiviral, also antibacterial response, whereas the ones that are overexpressed, inflammation, we heard that before, cell growth, differentiation, transcription, and you may say that's nice, but what does it mean? Well, Steve Cole's day job is to look at human with leukocytes, and when he published this paper with John Cacioppo some years ago, he found that the same pattern in people who describe themselves as lonely as opposed to socially integrated, you see as you see in the pure-reared monkeys. So I'm not about to argue that pure-reared monkeys are the same as lonely adult humans, but you have to note that their expression profiles are pretty similar. So very recently, we have introduced an intervention with our monkeys. The intervention involves when we put these groups of monkeys together at 7 months of age, we're now adding what we call foster grandmother and foster grandfather. The foster grandmother is a mother with a previous history of being very nurturing to her offspring. The foster grandfather has a previous history of doing well with kids. The grandmother is there to soothe those infants that need some help. The grandfather is there to break up fights. This is Doogie, one of our grandfathers. Nobody's going to mess with Doogie. And when we do that, we find that these changes, these differences, environmental differences, are reversible. So here are our data. These are methylation data from Moshe Ziff's group. Very, very quickly, at 1 month of age, in both males and females, there are 5,000 genes that are differentially methylated. The intervention comes here, and by 2 years of age, the number of genes that are differentially methylated have been cut in half for males and down to 2,500, and they're down to 750 in the females. So these patterns are reversible by environmental interventions. I'll skip this. This was to say that we have naturally occurring cases of neglect and abuse in monkey females. Their offspring look just like pure-reared monkeys, and I'll just leave it at that, so that maternal behavior can have an effect on how these monkeys grow up. And this pattern of mothering is transmitted from one generation to the next. Now, could we please have the video? Other ways that mothers differ is in their relative height position in the dominance hierarchy, and it has consequences for their offspring. So here is a video of a low-ranking mother with a 6-week-old infant. There's a fight going on off camera. What does the mother do? She can't afford to let that infant go check out the fight, so she grabs it. She grabs the flower. She distracts the infant. As the fight ends, now that the fight is over, the infant is free to go and explore in an area that she would not have the power to rescue it if it had gotten into trouble. Now, I offspring of a high-ranking mother could have just walked right into that fight, and that would have broken up the fight. Everybody would have scattered. So those infants have very different experiences. We now know that both the mothers and the infants have higher levels of low-ranking individuals, have higher levels of hair cortisol, they have higher incidence of infection and higher incidence of wounds from fights than their higher ranking counterparts. In the long run, as we follow these monkeys up, we expect to see some consequences for long-term mortality and morbidity. We also have data that suggests that if you look at the placentas of high and low-ranking females, they match the placentas in terms of their their methylation, genome-wide methylation pattern. They match the patterns that you see in their offspring at 2 weeks of age, and there are significant differences in the methylation patterns of high-ranking females and low-ranking females, and in their placentas that are mirrored in their placentas and transmitted to the next generation via whatever is going on in the placenta. So Rhesus monkey's dominant status is heritable, is passed on through epigenetic patterns. I was asked in making this presentation to talk about other species. I don't have time to talk about other species, given that big zero that we saw all over there, but just, oh, this stuff starts on first day of life, at face-to-face things. This is our first infant this year. See the snow, and already on day one, you're getting that face-to-face interaction. All of these behavioral patterns can be transmitted and are very different from whether individuals are high-ranking and low-ranking. And then finally, I talked about some of these alleles before. It turns out, with respect to, say, the serotonin transporter allele, Rhesus monkeys have both the long and short, but you look at other macaques, like Barbary macaques, they only have the extra long version. If you look at pigtail macaques, they only have the long version. If you look at Tonkanen macaques, they only have the long version. If you look at crab-eating macaques, they only have the long version. If you look at sun-tail macaques, they only have the long version. If you look at Tibetan macaques, they only have an extra short version. What's different about these Rhesus monkeys from all of these? Well, what's different is they have a polymorphism. And if you look at the other species of the other macaque species, they not only do not have polymorphisms in these genes, but they don't have them in the other candidate genes I mentioned. Great apes don't have these polymorphisms. Baboons don't have these polymorphisms. So what's different about Rhesus monkeys and humans than all these others? And the difference is they can live anywhere. And maybe, just maybe one reason underlying their success as a species, and perhaps ours, is not some kind of genetic specialization, but it's genetic diversity. Thank you for your patience. And these are some of the people that did it.